Hi, and welcome to the Sound of Science. Today on the AP edition, we're going to be talking about surface area to volume ratios and the cell membrane and how all of those things are related. What do we mean by surface area to volume ratio? And we talk about it a lot in biology, and I know that it's very important in biology, but why? So the amount of surface area there is on the outside of a structure per the amount of volume there is inside really determines how efficient the structure is at moving materials where they need to be. So when we talk about moving things like oxygen in or waste out or nutrients in or monomers out or whatever it is, the larger the cell is, the more demand there is for items, but the less surface area there is to provide access to those items. So larger cells are less efficient. This is why it's important that structures remain small. Usually that larger ratio is much more efficient than a smaller one, and we see that with smaller structures or smaller cells. So I'm using the ubiquitous gelatinous cube here to explain this large gelatinous cube here. We're going to have a difficult time getting access to things that are inside the center of this cube. It's got a long distance to go to get to the outside. Whereas here in this small gelatinous cube, we could just practically reach in and grab out whatever weapon, shield, whatever we wanted from this cube. The same thing goes with anything that we throw at it. It doesn't have as far to travel to the center of the cube in the small one, but on the large gelatinous cube, it's going to take a long time for things to move to the center of the cube. If we're not talking about a mythical monster and instead we're talking about a cell, this can be the difference between a cell's ability to survive or not. How do cells handle this challenge of maintaining a good, efficient ratio? Well, membrane folding. Membrane folding is the secret to survival when it comes to living things. So internally, we see things like mitochondria and chloroplasts. They need lots of surface area to perform their processes. In fact, the membranes, the inner membranes, are some of the most important parts of both of those organelles. Externally, we're going to see things like root hairs. So root hairs increase the surface area of plant roots. Plants need to be able to absorb lots of nutrients from the soil, lots of nitrogen, right? And lots of water. In order to do that, they need lots of surface area to be able to move it in. So in order to increase that surface area, instead of making the roots smaller, it adds these little projections that come. Another way that cells are going to handle this challenge, or how organisms handle this challenge, is the use of intestines. So intestines are pretty important. We actually use that to digest our food. We use intestines to pull monomers out as well as nutrients to move them into the bloodstream where they're needed so that we can get access to all of that energy. So as they are allowing food to move through, intestines actually have these things called villi, and they're just little finger-like projections that stick out along the inside of our intestinal wall. The villi themselves actually have further membrane folding with microvilli that stick out from, you guessed it, the villi themselves. All these little projections actually allow our intestines to be much more efficient. And finally, along that same line, other specialized structures that can aid in dissipation of things like heat or gas exchange are things like elephants using their large ears. This is a picture of an African elephant. African elephants living in a much warmer climate have evolved these giant ears. And their giant ears are not going to serve more as like a fan to catch the breeze, but instead it actually increases the surface area to allow for heat to dissipate from the animal. In addition, plants, because so much of their broad leaves are covered with cuticle, with that layer of lipids that is going to prevent the gas exchange, and also prevents it from drying out. Leaves are actually going to be equipped with these specialized stomata that open and close to allow for gas exchange. Let's talk a little bit more about the cell membrane itself, the structure of the cell membrane specifically. The whole point of the cell membrane is to create this boundary between the internal and external areas of the cell. It's going to control the transport of materials into and out of the cell, kind of like a bodyguard, right? That's its whole job. We know that the cell membrane is semi-permeable. Some things can move through and some things can't. The membrane itself is actually made up of lipids, specifically phospholipids. There happen to be amphipathic, which just means they're both hydrophobic and hydrophilic. And so when you look at them in an aqueous solution, these phospholipids automatically form layers. They actually sort of attach to each other and they form this bilayer with our hydrophobic tails on the inside pointing towards each other and our hydrophilic or water-loving phosphate heads on the outside towards both the cytoplasm environment on the inside and usually an aqueous solution on the outside of the cell. There are often proteins embedded in the membrane as well. So we've got these phospholipids in a bilayer, and then we have these embedded proteins. So there's two types we're going to talk about. The first is the peripheral. The peripheral membranes are the ones that are sort of loosely attached to the surface. They're hydrophilic. They have charged polar side groups. They're just sort of hanging out doing their thing. And then we have our integral membrane proteins. And if you look at the picture, you can actually immediately see the difference. Integral is integrated throughout the phospholipid bilayer. So they span the membrane. Hydrophilic, some of them are going to be hydrophilic with charged polar side groups, and some are going to be hydrophobic with nonpolar side groups. These are the ones that are going to go all the way through the interior of the bilayer. The example we would have here would be like a transmembrane protein, usually used for things like transport into or out of the cell. The proteins that are found in the cell membrane have a lot of different functions. And so to start with, we've got transport, and that's transport into or out of the cell. Cell-to-cell recognition. Cells actually have to recognize each other, and this is part of how your immune system works. Enzymatic activity, signal transduction, that's cells communicating with one another, intercellular joining, or the attachment for extracellular matrix or the cytoskeleton. So why is the fluid mosaic model fluid? Well, the thing is the phospholipids and the proteins move fluidly. They're not static. It's not, we look at these models, we can actually understand that these are constantly moving around each other. Cholesterol found in your membrane layers actually helps with this, and they help regulate how fluid the membrane is, in particular, depending on temperature. Carbohydrates and lipids, due to the diversity that we find in these molecules, can actually serve as markers for cells. And so we'll find those attached to the cell membrane as well. Two types, glycoproteins, which are carbohydrates that are attached to a protein, and glycolipids, which are carbohydrates attached to a lipid. So that's all we had time for today. Make sure that you understand this fluid mosaic model. You will see this model again, and make sure you understand all the components to it and why the cell membrane works the way it works. In our next video, we're going to talk about cellular transport and how things move in and out. For right now, I'll see you next time.